Previously, in our introduction to the field of biotechnology, we mentioned a specific type of molecule known as a restriction enzyme or restriction endonuclease. Now, restriction enzymes are these natural biological molecules found inside many prokaryotic cells, such as bacterial cells. And what the biological purpose of these molecules is to protect those cells from different types of foreign pathogenic DNA molecules. For instance, if a virus that carries DNA infects a bacterial cell, the bacterial cell can protect itself by using these restriction enzymes. And what these restriction enzymes do is they bind onto that viral DNA molecule and they cleave it, they cut it up into smaller, non-functional fragments. Now, biochemists can actually extract and clone these restriction enzymes from these bacterial cells and prokaryotic cells, and we can use them for a variety of different purposes, as we'll discuss in the next several lectures. So there are 4 purposes I've listed on the board. So we can use them to analyze and study some DNA molecule that we're interested in. We can also actually use them to determine the actual sequence of nucleotides of very, very long DNA molecules. So if we have a long DNA molecule, we can use these restriction enzymes to cut the molecule into smaller fragments, and then we can determine what the sequence is of the smaller fragments, and by piecing together, by determining the order of the fragments, we can then determine what the order of the sequence of nucleotides is in that long DNA molecule. Now, we can also use restriction enzymes to create recombinant DNA molecules, and then we can actually clone and amplify those molecules if we want to. And there are many other purposes, as we'll see in the next several lectures. Now, let's move on to the mechanism of action. How exactly does this cleaving process take place? So, restriction enzymes are these proteins found in these natural biological system cells, and they cleave via the process of hydrolysis phosphodiester bonds that hold nucleotides together. And these restriction enzymes basically cleave 2 phosphodiester bonds in a single double helix DNA molecule. And they cleave at specific palindromic sequences. Now, to see what we mean by all that, let's take a look at a specific type of restriction enzyme that is found in E. coli bacterial cells known as EcoR1. Now, this restriction enzyme basically cleaves at this location and this location along the following palindromic sequence. So notice we have a double-stranded DNA molecule, we have one strand, and then we have the complementary lower strand. And notice the blue section basically describes the palindromic sequence where this EcoR1 actually cleaves. So we have GAATTC, and then if we read this way, we get also GAATTC. So palindromic simply means if we read it this way, it will be the same as if we read it this way along the complementary strand if we read it in this direction. So we go from 5 to 3 on this strand and from 5 to 3 on this strand. And so it reads the same exact sequence. And notice that it cleaves between the GA on this side, and that means it will cleave between the GA on the opposing palindromic sequence. So this and this is where it will use the process of hydrolysis to basically break the phosphodiester bonds. And so this is one phosphodiester bond and a second phosphodiester bond that it will break. And so once it cleaves those 2 phosphodiester bonds, we basically produce the following 2 fragments of DNA. And notice in this particular case, we have these staggered or uneven regions that we form, and these are commonly known as sticky ends. Because if we stick them back together, because they're complementary, they will essentially form those hydrogen bonds, and so we reform that strand that we essentially broke this into. So notice that the restriction enzyme cuts at a palindromic sequence shown in blue. The cut sites are symmetrically positioned along the two strands of DNA. So we cut this region between the GA and this region also between our GA. Now, another way of saying that our mechanism of action is along a palindromic sequence is to basically say that the way we cut our double-stranded DNA molecule is to form a molecule that is characterized by a twofold rotational symmetry. So this should be rotational symmetry. So one interesting property of the majority of the cleavages that take place is that they are characterized by a twofold rotational symmetry. Now, to see what we mean by twofold rotational symmetry, let's take a look at the following diagram. So let's suppose we take just this blue sequence. So we have GAATTC and then we have CTTAAG. So this side we're going to label in black. So we have 5' and we have 3'. This side we're going to label in purple. So we have 3' and we have 5'. So we add our EcoR1 restriction enzyme and it cleaves. It breaks the phosphodiester bond here and a phosphodiester bond here. And so we produce the following two fragments with these two sticky ends. So what do we mean by a twofold rotational symmetry? So what we mean by that is if we take this molecule and we rotate it first about the x-axis 180 degrees and then about the y-axis also by 180 degrees. So if we make the two types of rotations a twofold rotation, then at the end we will produce that same molecule. So to see what we mean, let's actually conduct our rotation. So we make the cleavage, so what we get is one fragment contains the 5' end in black with the G and the 3' end, so CTTAA. And then the other end contains the 3' AATTC and the 5' in purple given by G. So if we make the first rotation by 180 degrees in the along the y-axis, so we basically flip it this way, these two will end up on this side and these two will end up on this side. So the 3 will end up here, the 5 will end up here, the black 5 will end up here, the black 3 will end up here. And so will these corresponding sequences. So here we have CTTAA, here we have G, here we have G, and here we have CTTAA. And now if we rotate this fragment 180 degrees along the x-axis going this way, we essentially flip it this way. Then what we get is the 3 will go on the bottom, this 5 will go on the top, this 5 will go on the bottom, this 3 will go on the top, and so will these sequences. And notice what we get. After the twofold rotation, the 180-degree rotation about the y and the x-axis, we essentially produce a molecule that is exactly like this molecule here. And that's exactly what we mean by a palindromic sequence. So this is what we usually see when we're dealing with these restriction enzymes. When restriction enzymes actually cleave the double-stranded DNA molecules, they cleave it in such a way so that they produce this twofold rotational symmetry. And what that basically amounts to is they cleave at palindromic sequences as the one described in this particular example.